Ladies and gentlemen, welcome and thank you for joining today's Heartwater disease overview. Please note that all lines will be muted until the Q&A portion of the call. We will provide you with instructions on how to ask a question at that time. You are welcome to submit written questions during the presentation, and these will be addressed during Q&A. To submit a written question, please use the chat panel on the right-hand side of your screen and choose All Panelists from the Send To drop-down menu. If you require technical assistance, please send a note to the event producer. With that, I'll turn the call over to Liz Clark. Please go ahead.
Good morning, everyone. I'm Liz Clark with Professional Development Services, and I'd also like to welcome you to today's webinar. We have 4 speakers today. Our first speaker is Dr. Beto Perez de Leon. Dr. Perez de Leon serves as Director of the United States Department of Agriculture, Agricultural Research Service, Nippling Bushland U.S. Livestock Insects Research Laboratory, leader of the Livestock Arthropod Pest Research Unit, and co-leads the USDA ARS Veterinary Pest Genomics Center. He spent 10 years working in the private sector researching and developing animal health products before joining USDA ARS. Dr. Perez de Leon obtained a degree in veterinary medicine from the University Veracruzana in Mexico, Master of Science from the University of Georgia, and a doctoral degree from the University of Wyoming. He also has adjunct faculty appointments at North Carolina State University and Texas A&M. Our second speaker is Dr. Robert Miller. Dr. Miller received his PhD in entomology in 1997 from Kansas State University. He is currently a research entomologist for USDA Agricultural Research Service, and his work is to develop new strategies to control cattle fever ticks, especially those that are resistant to pesticides. Our next speaker will be Dr. Sari Rivera Garcia. Dr. Rivera is an epidemiology officer for USDA APHIS Vet Services in San Juan, Puerto Rico. She received her DVM from the University of Missouri and later completed an internship in food, animal, and production medicine and a Master's of Veterinary Public Health at the same institution. From 2009 to 2012, Dr. Rivera worked at the University of California Davis Veterinary Medical Teaching Hospital as a resident veterinarian, where she treated a variety of livestock, including dairy, beef cattle, bucking stock, sheep, goats, and swine. In 2013, she joined the USDA as a supervisory public health veterinarian with the Food Safety and Inspection Service, where she regulated small and large meat-producing establishments in the states of Pennsylvania, New York, Vermont, and New Hampshire. Dr. Rivera joined USDA APHIS Vet Services in 2016 and currently works with swine, cattle, avian, and small ruminant health programs in Puerto Rico. And our last speaker is Dr. Fred Sotera. Dr. Sotera received his DVM from Kansas State University in 1981. He joined Veterinary Services as a field VMO in 2002 after owning a small animal and exotics practice in Puerto Rico and serving as the veterinarian and director of the Puerto Rico Zoo for many years. He became a foreign animal disease diagnostician in 2003 and started working with the swine industry in Puerto Rico and Dave Pyburn from the swine staff in classic swine fever surveillance and the illegal boat landing program to mitigate the international waste products that were arriving in small boats from the Dominican Republic and Venezuela. Dr. Sotera is involved with the swine staff in the CSF surveillance program in Puerto Rico that targets the garbage feeder producers as a high-risk activity for transmission of swine diseases and an active surveillance in Mona Island with feral swine. With that, I'm going to turn it over to Noella Moyeno, and Noella is going to explain a little bit more about the webinar.
Thank you, Liz. So, just to give you an overview of what the webinar will be, our purpose for the webinar is to provide an overview of the Heartwater Etiology and Ecology to all participants, and this is in preparation for a tabletop exercise in San Juan, Puerto Rico, which is scheduled for next month, June 2018. Several subject matter experts across the USDA, as Liz previously introduced, will be providing this webinar presentation. And to give you a little bit of the agenda that we'll be going through today, Dr. Perez de Leon will be giving the overview of Heartwater, basically the history of Heartwater and the current the status here in the Caribbean. Dr. Miller will follow, talking about etiology and transmission and basically the vector of Heartwater disease. followed by Dr. Rivera Garcia, which will be talking about pathogenesis, clinical findings, and how to diagnose the disease. And finally, Dr. Soltero will be talking about treatment, control, and preventative measures that are currently established not only here in Puerto Rico, but in the Caribbean. I would like to mention Dr. Denise Bonilla was scheduled to provide the portion of treatment, control, and prevention. Unfortunately, she was not able to be with us today. So Dr. Soltero will be providing that portion of her presentation. And with that, I will continue to pass this presentation to Dr. Perez de Leon.
Hello, Dr. Molino, and thank you, everybody. Do I have the rights now to move on through my slides? And can you hear me? Okay, now I am the presenter. Thank you. Again, thank you, everybody, for attending this very important webinar. And I say that because if you were not aware of it, we have the emergence of an exotic tick species in the continental U.S. that is Hemaphysalis longicornis, native to Asia, and all of a sudden emerged in New Jersey last year. And earlier this month, I guess, or late last month, we were notified that it has been detected in Virginia. So that event, a significant event for me, if you will, provides a very important context for the topic that we are discussing today. And again, I'm going to go over disease history and endemicity in the Caribbean for Heartwater and its tick vector that is present, that we know that is present in that part of the world, in the Caribbean region. And hint, of course, we're talking about a tick-borne disease. And I would also like to stress that it's important to consider this as a tick-borne disease system because ecology plays a very important part in terms of where the tick is, the vector, and the epidemiology of the disease as well. That's why we're going to talk about not only the disease agent, but the tick vector as well. And then we're going to put all that information for this portion of the presentation in terms of the risk for U.S. entry and what has been done so far and what needs to be done to address or to adapt that preparedness to come up with countermeasures to prevent such an event happening here in the U.S. primarily, but in the continental Americas for that purpose. So the next slide talks about, we're going to start with a brief historical account. And it's interesting because as I was preparing these, I did it chronologically, and you will see that the first bullet there relates to the vector itself for how we are addressing this topic today, and that refers to the arrival or when it was first documented that Amblyomma variegatum, that is scientific name for what we know commonly as the tropical bont tick. 1830 is when we have the historical record for its introduction to the Caribbean from West Africa. Of course, through infested livestock, and we believe it was cattle that was brought in from that part of the world into the French Antilles, and we'll get into that in more detail a little bit later. After that, 8 years later, in 1838, we have the first report. And one more thing, you will see that at the bottom of most of my slides I have references. We have most of those references, if not all, available. And if you are interested in getting a PDF of those references, please contact Dr. Molino. We will have available to the attendants. Again, NINTAS was the name ascribed locally to what we believe now is an initial reference to what we know now as Heartwater, and that was done in South Africa, and that was related to disease detected in sheep 3 weeks after a massive tick infestation in that part of the world. And then by this triggered, as you can imagine, research by the colleagues in South Africa who have kept very relevant historical records. And by 1888, we already had the name of Heartwater coined for this livestock disease that, as far as we understand now, is indigenous to Africa. But this is another important aspect to keep in mind because, as I mentioned, these tick-borne disease systems have evolved through hundreds of thousands, if not millions of years. They tend to be zoonotic in some cases, but certainly, generally speaking, evolved initially in a cycle that involved wildlife and then jumped into livestock through the domestication of different animal species. And that's one of the reasons why we study them, because of the impact on not only animal health, but public health as well. Next slide, please. Then in 1998, Dixon and Eddington separately were able to produce Heartwater artificially by intravenous inoculation of blood from sick to susceptible animals. And in 1900, Lounsbury confirmed the suspicion at that time in South Africa that the bont tick, this is another Amblyomma species, Hebraeum, was reverted to South Africa in that specific part of the continent. And there are a number of vectors in Africa, or tick species belonging to the genus Amblyomma that can serve as vectors of the causative agent of Heartwater, and I believe Dr. Miller will touch on that. I am not going to steal any of his thunder. But by 1925, Dr. Cowdery documented the presence of rickettsia that he named Rickettsia ruminantium in tissues of affected animals and in infected ticks. And this is very interesting because in the history of science, Cowdery was a Canadian rickettsiologist working with the Rockefeller Institute for Medical Research in New York, who had been invited by Taylor to investigate Heartwater in South Africa. And then in 1930, Dobney, working in Kenya, he noted an outbreak of Heartwater, and through his research, he documented for the first time that the tropical bont tick, Amblyomma variegatum, was a biological vector of Heartwater. By 1947, excuse me, the name of the causative agent was changed to Caldria ruminantium by Moshkovsky. However, through phylogenetic analysis in 2001, Dumbler et al. Renamed Heartwater or the Heartwater agent as Ehrlichia ruminantium. That's how we know these microbes of today. Continuing with the historical account, in 2015, Collins et al. Published the genome sequence of Ehrlichia ruminantium, and we believe that that will that has opened up exciting opportunities for control measures based on the pathogen against this disease. But by 2017, where we are now with our understanding of the endemicity of the disease in the Caribbean, according to Gondart et al. In their NICE review published in 2017, Heartwater remains endemic in the islands of Antigua, Guadeloupe, and Maya Galante. And just an update, the World Organization for Animal Health recognizes Heartwater as a not notifiable disease in the category of multiple species, diseases, infections, and infestations. So, remember I mentioned to you that by 1830, it was recognized that variegatum had been established or had arrived in some islands of the Caribbean. And by 1967, Morel suspected that Heartwater was present in or prevalent in Guadeloupe due to symptomatology similar to that seen with clinical cases in Africa, and of course the presence of the tropical bont tick vector. And as early as 1977, Willenberg pointed the serious threat of Heartwater to American livestock and the danger of its introduction to the American mainland. And by 1980, Perrault et al. Diagnosed Heartwater in Guadeloupe. So, Here in this next slide, addressing the threat of Heartwater in the tropical bont tick in the Caribbean, I succinctly mentioned what has been done to address this situation in this part of the world. We understand that the tropical bont tick, the disease vector, in this case the Amblyomma species, started to spread rapidly in the region in the late 1960s. By the end of the 1980s, 18 islands were infested, and it's suspected that a driver for this dispersal is the establishment of the cattle egret, or several cattle egret populations throughout the Caribbean islands. And several efforts were conducted to eradicate or to try to achieve progressive control of the tropical bont tick in the region with variable levels of success. For example, in Puerto Rico, colleagues from the USDA in the past were involved in that effort and were able to eradicate the tropical bont tick from Puerto Rico, and I believe Dr. Solfero will talk about that in more detail later. But again, the threat remains, and here I am showing a fact sheet that APHIS, the Animal Plant and Health Inspection Service agency, our sister agency of the ARS, Agricultural Research Service, put out in 2013, where they listed high consequence foreign animal diseases and pests, and you can see that they took a tier approach, and Heartwater is listed in tier 2, and the tropical bont tick in tier 3. That fact sheet is available through that link or that web address shown at the bottom of this slide. And just so you know, really quick, 8 criteria helped guide the decision on whether or not designate an animal disease or pest as one of high negative consequence. 1, high epidemic potential, or the ability to rapidly spread and infect a large number of animals. 2, high economic impact. 3, large impact on trade, both domestic and international. Number 4, high animal morbidity and mortality, or the capability to cause disease and death, respectively. Number 5, high potential to infect multiple species. 6, inability to detect the disease rapidly. 7, ability to vaccinate for the disease. And 8, high zoonotic potential. The next slide, we provide some examples of how Heartwater and the vector present a threat to the U.S. mainland. This is a summary of information presented by Casari et al. In that reference shown at the bottom of the slide. And again, a factor that could be involved is migrating cattle egrets carrying immature ticks. Ticks introduced through domestic and livestock from the Caribbean, symptomatic carrier hosts, increased imports of exotics, transport in feed, bedding, fomites, etc., high mortality observed in experimental infected white-tailed deer, additional issues where exotic ranching occurs, vertical transmission in cattle, and in this regard, previous researchers have documented that native species of some, or at least Amblyomma maculatum, which is a native species of Amblyomma in the continental U.S., is a competent vector for Ehrlichia ruminantium. So very important aspects to keep in mind in perspective considering this threat. And then here we summarize data from a publication by Dr. Burrich et al. In 2000, providing a real-life example of the threat, where they showed that Amblyomma sparsum was infesting leopard tortoises imported from Zambia, and all of the, or 40% of the Amblyomma sparsum ticks were positive for Ehrlichia ruminantium by PCR. However, the Amblyomma marmorium, none of them, fortunately, were positive. All of them were negative. But again, how did this happen? We don't know exactly, but the infection detected by PCR likely indicates that the ticks were infected during the immature stage while feeding on a host, it could be buffalo, while they were still in Africa. And then all those ticks arrived infesting those tortoises in one shipment from Zambia that was sent to Central Florida to a reptile importer. And then to go back to the future, if you will, this is the last slide in my section. I show what I think is a great concern and present concern, certainly based on this, the results from this publication by Variegatum et al. From 2016, where they were able to document that Rhipicephalus microplus is susceptible to infection by Ehrlichia ruminantium. And you will see that this was detected in West Africa. Rhipicephalus microplus became invasive in West Africa recently. And now you have this other dimension with the threat for the arrival of, potential arrival of Heartwater in the Americas, mainland American continent. So that is the last slide in my portion, so I'll turn it over to Dr. Miller. Thank you.
Okay, good morning. So today we're going to talk about the tick. A little bit about, first we'll talk about the identification of the tick, the host range. We'll move into life history and reproductive capacity. We'll go over agricultural, human, and economic importance of the tick, how to survey for the tick in the environment to learn about its population or its presence at least, and control of the tick. And then we'll finish up with the genome project that we have with the USDA in collaboration with CRAD. Okay, key characteristics to the identification of Amblyomma ticks. Now, this is Amblyomma is the genera of ticks that incorporate many different species. One is they have very long mouth parts and very long palps. So the palps are long, and segment 2, you can see the number 2 right there. That segment 2 is at least twice as long as it is wide, and that's a key characteristic that differentiates these ticks from other hard ticks, such as exotic ticks or Rhipicephalus ticks. They have their mouth parts are shorter. They're generally ornate. This means that they're very colorful ticks in general. There are 1 or 2 species that aren't super shiny. They also have really, and there's some pictures in following slides, but they have really neat geometric designs on them. So they're very easy to differentiate between other ticks that are just maybe brown or kind of a reddish color. They have eyes. If you look at the diamond-shaped area, those round circles on the diamond-shaped area are their eyes. They have festoons. I'll point out the festoons a little bit later. That's on the abdomen. They're little square designs along the border of the abdomen. The basis capituli is variable, but it's usually subtriangular or subrectangular dorsally. So below the palps, there's a little triangular area with the 2 dark spots on it. That's the basis capituli. And in these ticks, these Amblyomma ticks, they're kind of this rectangular shape. You can see that kind of rectangle. And there are other ticks, other tick genera have different shapes. Some are square, some have different kind of lobes on them. So that's something you can look at and tell these ticks apart pretty quickly. We have at least 8 species of these ticks in the U.S., 4 of which are important livestock pests. And there's 5 species frequently found on livestock and in Africa. Okay, so in Africa, members of the genus Amblyomma are called bont ticks, and the word bont is Afrikaans. Afrikaans is a mixture of Dutch and African native languages, but it refers to the presence of brightly colored patterns on the scutum and the white banded legs. So you can see that these are very beautiful ticks. You can see that they're very ornate. They have those geometric patterns on their scutum, and then they have these really, really beautiful stripes on their legs. Now, the U.S. Amblyomma do not have these banded stripes on their legs, and we have Amblyomma pathogenesis and Amblyomma imitator, that have these really nice geometric colors and shapes on their body, but they don't have the stripes on their legs, and the pattern of these geometric shapes and colors is different. So it's pretty easy to tell these ticks apart from the ticks that are native in the U.S. Okay, so this tick has a very wide host range. Common hosts for the adult ticks include cattle, sheep, horses, goats, antelopes, camels, and a long list of large wild animals. Basically, any ungulate can be fed on by these ticks. Humans have also been reported as hosts for the adult stage, so it does attack humans. Nymphs feed on a great variety of mammals, and medium to large size, and occasionally man. And here's where birds become important. Birds are frequently attacked by the nymphs. And again, so there's 3 stages of a tick. The egg hatches, and there's a 6-legged larvae, and then that larvae feeds, and then after it feeds, it molts. It sheds its skin and becomes a larger tick that has 8 legs. And that's a nymph. It's not an adult. It's not reproductively active. It's kind of like a sort of pre-teenager. And then that nymph feeds and molts and becomes the adult. And that's when you have differentiation between males and females. You have sexual reproduction, and the females lay eggs. I add these 2 pictures here. This larvae is a questing larvae. So ticks have Haller's organ on their front legs, and that organ detects CO2 much like a mosquito uses its antenna to detect CO2. When they sense an animal coming through vibrations and CO2, they stick their legs out and they grab a hold of the animal as it comes by, and that's how they infest them. And then this is a cattle egret. Cattle egrets were introduced into the Caribbean in the 1950s, and if you remember from what Beto said earlier, the rapid spread of these ticks to the Caribbean was started around the 1960s and has continued through by the 1980s. They had spread to these 18 different islands. So if you think that the egret presented itself in the Caribbean in the 1950s and had time to reproduce and start to spread around, they're obviously spreading the ticks around as well. Banded egrets have been, banded in the Caribbean, have been found in Vermont and also in Florida, so they do travel to the U.S. Okay, a little bit of the life cycle of the tick, getting back to the tick. This following information is taken from Hoogstraal, 1956, and applies to Sudan. Harry Hoogstraal was a Navy entomologist, a civilian Navy entomologist, and was super prolific in writing about ticks in Africa. So here's what Hoogstraal says. Adults appear towards the end of the dry season, first males and then females. Populations increase in numbers and remain high through the rainy season and decrease rapidly in the dry season, although a few specimens may be found even then. Larvae and nymphs gradually become more numerous in the dry season, while some nymphs are found during the rain, are scarce. So you can see how the larvae and the nymphs are taking advantage of the smaller animals that can handle the dry season. And then when the wet season comes and the larger animals come back and the grass is there, the adults are taking advantage of the adults. So in areas where there's only one rainy season each year, there's only one life cycle, or one generation is completed a year, or one life cycle is completed a year. In areas that have 2 rainy periods, there are some regions in the tropics that have 2 rainy periods a year. Those areas will have 2 generations per year. And next slide will give you a little bit of explanation of why this would occur. So we'll go over the life cycle. The Amblyomma variegatum is a 3-host tick. So this means that when the egg hatches and the larvae come out when they're in the grass, they're going to get on an animal, they're going to feed, and after they finish feeding, they're going to drop off the animal, they're going to molt to an nymph stage, then they're going to go and look for another animal. They'll feed on that second animal, and then they'll drop off, and they'll molt, and they'll find a third animal. So it's a 3-host tick. Other ticks, like Rhipstocephalus microplus, the cattle fever tick, is a 1-host tick, and that tick, the larvae gets on an animal, an individual animal, and will not drop off that animal. It'll go through the larval, nymph, and adult stage on a single animal. That's a single host tick. So these are different. These are 3-host ticks, and that's important because when you try to control these ticks, it changes things. So one host tick is relatively easy to eradicate or control because you just dip the cattle that they're on and you're done. These ticks are getting on all sorts of little rodents and birds and skunks and raccoons and anything that it can get on, and then also gets on wildlife and domestic larger animals too. So it's difficult. The females are large. These are very large ticks. The female lays 10,000 to 12,000 eggs. The pre-oviposition period, so meaning after she's replete and she falls off and she crawls under a rock and digests her blood meal, it takes her 12 days to start laying eggs. After the eggs are laid, it takes 53 days to hatch. And then when the larvae and the nymph feed, so when they find an animal and they start to feed, it takes them 5 days to feed before they drop off and then begin the process of development and molting to the next stage. A female will feed for 12 days. She has to feed a little bit, go through some physiological development, and then she needs to mate on the animal, and then she needs to go through a rapid engorgement. That's why that's taking a little bit longer. So it takes 141 days. If you want to be quick about it, between all the developmental periods and all the feeding periods, it takes 141 days. So if you remember from the last slide where we have the environmental factors that really affect the survival of these ticks, when you have 2 rainy seasons, approximately 6 months apart, you can get 2 generations per year. But if you only have one rainy season, you're only going to knock out 1 generation per year based on how long it takes for these different life stages to feed, and plus the environmental factors. Okay, a little bit about the agricultural importance of this tick. The bite of the adult tropical bont tick is especially serious since the mouth parts penetrate very deep. Remember from that first slide, they have these really long mouth parts and they drive very deep into the animal. This results in the formation of wounds and abscesses. Severe lameness may be caused by the attachment of the ticks in the interdigital class of animals. A definite reduction in the value of hides and skin is caused by heavy infestations of Amblyomma variegatum. That's because there's a lot of scar tissue, and so when you try to make a nice piece of leather to make a pair of nice shoes or a nice jacket, you don't want all these scarring inside the leather. You want a nice clean piece of leather. So the cheaper leathers are going to have all these scars in them. In some areas, the larvae are serious pests of humans, and attached on the legs and around the waist. Larvae reportedly may burrow under the skin and cause intense irritation and inflammation. So you think about having this tick that's just burrowing underneath your skin, especially along your belt line, your waist. It's got to be very painful. A little bit about the economic importance. Given the highly suitable conditions for this pathogen in the United States, the pathogen is predicted to spread rapidly after emergence and cause up to $760 billion in annual economic losses. Now that $760 billion is probably a few years old, so within the last probably within the last 10 years. So you got to, with the inflation and things, it'd probably be a little bit higher. And let's look at why it's going to be such a high number. So this map shows the potential bont tick distribution in the United States, and you kind of say, well, okay, that's not that much, Gulf Coast, from Texas to Florida. But we also have an endemic tick called the Gulf Coast tick, which is Amblyomma maculatum. And if you look at that host range in the tan, that covers a good portion of the southeast United States, including some pretty big cattle producing parts of our country. And that dip to the south, below Tennessee, is I don't know how real that is. I'm not sure if there's such a difference in climate there that would account for that. It may be possible that the tick does occur straight across, Tennessee, probably the lower half of Kentucky into West Virginia and on over towards and to meet up with the area below Pennsylvania. So we have to do some surveillance and make sure that the Gulf Coast tick is not in those locations. So the importance of and Beto had mentioned this before, but in 2000, lab studies with goats indicated that the Gulf Coast tick was a vector of Heartwater just as efficient as the African Amblyomma ticks. So this is where we get the huge economic losses. Okay, a little bit about how we survey and control these ticks. So, of course, we want to inspect animals for ticks and speciate those ticks to make sure we know what species of ticks are out there. We also can flag for larvae, and flagging is you basically, if you see these gentlemen here in the picture, maybe it's one gentleman and one man, or gentle woman. You basically take a piece of flannel cloth and attach a pole to it, and then you have a string, and you just drag it across vegetation. And after a certain amount of length of dragging, you flip the cloth over, and you basically get on your hands and look for larvae and pull your tweezers out and try to get them. You can use CO2 and pheromone trapping to get at nymphs and adults. So there's lots of techniques to do this type of trapping. The ticks, again, are attracted to CO2 because warm-blooded animals are breathing out CO2, so the ticks are attracted to that. Ticks also have what's called aggregation pheromones, where they can put out these pheromones and the ticks will basically be attracted to each other and kind of clump together. So if you combine the CO2 with the pheromone, it's a little bit stronger attractant. If you notice, these people are also wearing kind of a suit made of flannel, and what they can do with that is as they walk through the brush, dragging their flags, they're also collecting, they can look at themselves and collect ticks off themselves. That's why you have a buddy and you check your buddy and then your buddy checks you. A little bit of control. Okay, so again, I've alluded to this before. Control is difficult due to the wide host range. It's hard to get every single little animal that's going to be a host for this. And in the Caribbean, some of the Caribbean islands, it's important to limit the need for water. There's not a lot of water around some of these places. So some of these pesticides that need water for mixing aren't going to be the best thing to use in every single situation. So you need to take account of that. So treating animals with a topical or systemic pesticide is a little bit better. Something like a pour-on, it's already mixed in the bottle. All you do is you pour it along the back of the animal, or injectable where you just, it's like a subcutaneous injection with a long persistence. A lot of these pour-ons and injectable formulations have very long persistence, and they're ideal for getting out in a very rural area with not a lot of water and not a lot of resources to get after some of the animals. Luckily, there's no known resistance to chemicals, at least in the Caribbean so far, in the experiments that have been done so far. So that's something that's beneficial. If the adults are getting on, domestic and larger livestock, one of the good things is there's in some of the islands in the Caribbean, you got sheep and goats. There's difficulty in finding pesticides that you can use to treat them, and you have maybe some cattle and a few other animals. Maybe have to, if there's some deer on some of the islands, maybe we have techniques for putting a systemic pesticide on corn that you can then feed to the deer. There's things like that where you can bait them in and treat them. Not an easy proposition, though. Okay, my final slide is the genomics work that we're doing in collaboration with CIRAD. And the objective of the genomics project is to facilitate the sequencing, assembly, and annotation of the genome and tissue-specific transcriptomes of the tropical bont tick. So far, our laboratory has worked with CIRAD and collaborated to generate genomic DNA samples of Amblyomma variegatum from different tissues that were from infected and non-infected ticks with Ehrlichia ruminatum. Genomic studies, genomic sequences have been obtained, and that was through the National Genomic Laboratory in Santa Fe, New Mexico. And then plans to assemble and annotate the genome are underway. Transcriptomic sequences remain to be generated as well. So the expected deliverables, the Amblyomma variegatum genome will be publicly available. Genomics research offers the opportunity to innovate safer control technologies that disrupt Ehrlichia ruminatum transmission to susceptible hosts by detecting the vector with anti-tick vaccines, RNA interference, and transgenics. So that concludes my portion of the talk. Thank you very much.
Okay, thank you. I'm just going to start with my part, which is basically all the more clinical side of things in regards to animals. So the pathogenesis of the disease, starting with inoculation, basically the pathogen is inoculated. It first infects, or not all of this pathogenesis has been completely elucidated, but this is basically the one that is most commonly proposed. The pathogen infects the reticuloendothelial cells, which are the ones of the mononuclear phagocyte system, so basically macrophages and the derivatives of those a lot of times. It does proliferate by binary fission within vacuoles of these cells, and then these cells degenerate and release the parasite. A parasitemia develops, and then the parasite infects endothelial cells, which is where it becomes mainly established and reproduces itself there as well. It causes a vasculitis, which leads to permeability changes in capillaries. And this is what actually leads to the multi-system pathologic changes that are usually seen. It is important in regards to inoculation. It not only can be transmitted by the tick, but it can also be transmitted vertically through colostrum or by IV inoculation of the organism. As far as clinical signs, there are different forms of the disease. The first one we're going to discuss is the peracute form. It is relatively rare. It is most common in, well, these and other syndromes or the other forms are more common in non-native breeds of cattle, sheep, and goats. And actually, the differences between native and non-native can actually be very significant. This syndrome, the peracute, causes sudden death preceded by terminal convulsions. And that can be preceded by respiratory distress, fever, hyperesthesia, lacrimation, and diarrhea. As far as this syndrome, it's actually common in heavily pregnant animals. As far as the acute form, it can show up as a fever, anorexia, listlessness, congested mucous membranes, and respiratory signs that lead to dyspnea, plus or minus diarrhea. CNS signs can also be observed, and these are sort of the ones that are most talked about a lot of times. They can show chewing movements, tongue protrusion, eyelid twitching, circling hypermetria, rigid stance, muscle tremors, and changes in behavior such as aggressiveness and anxiousness. As far as these signs, the CNS signs, they are more common in cattle and less seen in sheep and goats. During terminal stages of the disease, you can have severe neurological signs, which include lateral recumbency, paddling, galloping movements, opisthotnus, hyperesthesia, and nystagmus. They can have frothing of the mouth or seen at the mouth, and these animals usually die within 1 week. As far as the other forms, the last two are the subacute or mild or subclinical. The subacute form is fairly infrequent or not necessarily recognized because it is mild. They can show prolonged fever, coughing, and mild incoordination. And these animals usually either recover or die within 1 to 2 weeks. Finally, the mild subclinical form can be seen in young animals or those that are partially immune, that are indigenous or native wildlife, and it's usually referred to as Heartwater fever. As far as clinical pathology, what has been documented is basically, it's actually not that severe, at least not compared to the gross pathology lesions that are seen. They can show anemia, decline in thrombocytes, fluctuation in white blood cell counts, an increase in total bilirubin and hyperproteinemia. They can also have increased BUN and creatinine. As far as the postmortem findings, these can be quite severe and can show hydrothorax, hydropericardium, which is actually more common. The hydropericardium is more common in sheep and goats than it is in cattle. Hydroperitoneum, pulmonary edema, edema of the lymph nodes, splenomegaly, perirenal edema, and hemorrhages or petechial hemorrhages across different organs, including pleura, peritoneum, and the endocardium. And they can also have a hemorrhagic enteritis. The cattle, as opposed to sheep and goats, can also show, or more commonly also show, congestion and swelling of the abomasal folds. Continuing with postmortem findings as far as the central nervous system, the lesions seen include cerebral edema, microgliosis, necrotizing vasculitis, hemorrhage, microcavitation, and a focal necrosis of the granular layer of the cerebellum. If you look at the pictures to the right, you can see basically gross pathology in the brain and cerebellum, and basically a picture of the brain smear that basically can be done at the farm and sent into the laboratory. As far as differential diagnoses for these diseases, there are a lot of them. So things that can look like Heartwater include anthrax, rabies, babesia and anaplasma, tetanus, filariasis, trypanosomiasis, your bacterial meningitis, encephalitis, listeria, intoxications with different things, including lead and arsenic, and then heavy parasite infestations such as Haemonchus and coccidiosis. In that regard, nervous coccidiosis. As far as diagnosis of the disease, either pre-mortem or post-mortem, incubation period of the disease itself is 1 to 5 weeks. It has a mean of 2 weeks. Things that can be collected include biopsies for histopathology, which some of the tissues that are recommended or very important to be collected include the cerebellum, the cerebrum, and the hippocampus, and they recommend that you pick, if you are not sending entire organs or various tissues, that you select the most heavily vascularized areas. As far as diagnostic testing, PCR can be performed. It is usually performed in blood and bone marrow, but can also be done in brain, lungs, kidney tissues, and thoracic fluid. As far as serology, there are different tests available, including ELISAs and indirect fluorescent antibody tests, but these have limited applicability, and they can also cross-react with other Ehrlichia species. You can also, if Amblyomma ticks are observed, they can also be collected and sent. As far as sample submission, this is basically an excerpt from the FAD manual that it's given to us at the FAD course. Probably many people have this manual. NBSL basically recommends these samples to be collected, including serum, whole blood out of a purple top, a brain smear that has been air-dried and fixed, and both fresh tissues and formalin-fixed tissues, which should include, it can include different organs, but it should definitely include lymphoid tissues, brain, and kidney. This is my last slide. As far as treatment, the animals can be treated with oxytetracycline. The most commonly recommended dose is 10 mg per kg intravenously, twice daily for 3 to 4 days. However, some people use a higher dose at 20 mg per kg IV. Depending on the severity of the disease, it is very important that if given IV, these have to be given quite slowly. Long-acting preparations of Oxytet can also be used if the IV preparations are not available. Oxytetracycline treatment is beneficial or can be beneficial in the early stages, but a lot of times once animals have developed neurological signs, they die despite treatment. As far as recovery, animals that do recover are immune to homologous strains, but they're not necessarily immune to other strains. And there are several strategies in terms of control once the disease has been established, which basically involve some type of either a vaccination or a controlled infection tailored with oxytetracycline treatment. We won't go into much further detail into that today, as that should not be the case for us, or at least in regards to this exercise. So now I am going to transition to Dr. Soltero.
Okay, those of you that can see the map, as you see in this map, Puerto Rico and U.S. Virgin Islands are right in the middle between the Greater Antilles and the Lesser Antilles. And the proximity between the affected three islands right now, which is Antigua, Guadalupe, and Maria Galante, is very close. The cattle egrets can fly from one island to another, especially after hurricanes, they get moved around. And also there's a lot of merchant marine fishing vessels that can easily go from one island to another, apart from the normal merchant marine traffic that's occurring all through the islands. So, yeah, the high risk is always there. And there's an island between Puerto Rico and St. Croix, which is called Vieques, which we are using as an early warning disease system. We had in Puerto Rico and in Vieques Amblyomma in the 70s, which we had enough blood that we could eradicate it. We haven't seen it since. In the USDA on November 6, 2013, an adult male Amblyomma variegatum tick was found on a routine visit to a premise located on the western end of the island of St. Croix in Virgin Islands, U.S. Virgin Islands. This premise was the last farm waiting for release from quarantine after being treated with Coumaphos every 2 weeks for 3 years. The finding was reported to VSPR-AVIC, and notification to the Veterinary Services Leadership Team was performed. The index premise had a previous history of TBT during prior infestations on the island. The previous TBT finding on the island was in July 2010. And that premise was quarantined, and it, along with the surrounding premises, completed a 3-year protocol of treatments and follow-up surveillance in 2013. And this is because, as Dr. Miller said, the life cycle is so complex that treatments cannot be done on 1 year. It has to at least have a 3-year duration to fully believe that we can control it could come up anytime. So a 3-year protocol was established. USDA and Veterinary Services had a comparative agreement program in place for TBT, and there was a VMO and AHT on permanent positions at that time in St. Croix. A situation update, as of March 20, 2014, the number of premises positive is 1, and there's no change from that report since then. The number of premises quarantined is still 1, no change from that previous report. The number of animals quarantined at that time was 105, 32 bovines, 58 sheep, 13 goats, and 2 horses. This as of 3/7/14. Number of Amblyomma ticks found, 28. Species affected, sheep and cattle. The number of premises under surveillance at that time were 14, including the index premise. Total animals under inspection was 228 approximately, and there is a cooperative agreement between USVI and Veterinary Services in place until October 2018. Wildlife surveillance. It was conducted in 2015 by SCWIDIS, the Southeastern Cooperative Wildlife Disease Study in the islands of St. Croix, USVI, and in Vieques in Puerto Rico. They did extensive testing of mongoose and a little bit of bird surveillance, and no Amblyomma ticks were found in those studies. Currently, in Vieques Island, we have started CO2 traps this year, and will continue to do so as long as funding is available. There was an interstate movement ban from the Puerto Rico Department of Agriculture initially to protect movement from St. Croix to Puerto Rico, and which included all livestock from U.S. Virgin Islands. Then it was later modified to process and transport animals under seal by Veterinary Service 127 form that could come from USVI to Puerto Rico and come in through escort and quarantine to do take quarantine in Puerto Rico and then gradually or move to interstate to Florida. These PIDA restrictions remain in place pending an audit and final status of the affected index premise. There was an assessment team that was formed from District 2, a 3-member team that deployed to St. Croix from May 19 to the 23rd in 2014 to assess the current situation and determine the adequacy of controls to eradicate activity on the island. The team was composed by Dr. Jim Miller, which was the EP of South Carolina at that time, Dr. Noelia Moyeno, the CLVMO and now the Emergency Coordinator for Puerto Rico, and Israel Gonzalez, the AHT that was had experience in the Virgin Islands. They did recommend after their report to use Dectomax, which is an injectable product that is currently being used in Texas, to use RFID to control the movements and inventory, and to use the use of a spray box to improve the spraying of the animals. The current status right now, the Amblyomma ticks, the last time they were found was in 2014. No reports of Amblyomma ticks have been present since then. No reports of additional premises being affected. There has been no clinical signs of Heartwater disease in small ruminants or cattle. There is a cooperative agreement between VS and the USVI signed in March 2014 to provide extra funding for equipment and supplies to combat TBT. This year, more surveillance by squiddies is being planned, and there is a program audit that is scheduled for October in 2018 by the emergency staff, Julie Galpierre, VMO, and Denise Bonilla, the VS entomologist. For future projects, Veterinary Services will be participating in a CARIBET meeting coming up in Antigua next month to strengthen partnerships and assist with issues that are present in the region, and this is one of them. One of the topics to be discussed is ticks and tick-borne diseases, which concerns with concerns of tick resistance developing already in the region. There is a gap analysis workshop on Heartwater that Dr. Cyril Gay, which is USDA ARS scientist, that's being scheduled for October 2018 by the CARIBET Leadership Group. This is a great opportunity to have up-to-date information on the disease status of the region and ways to collaborate on treatment and prevention efforts. I just got this scientific report of the analysis of Amblyomma surveillance data in the Caribbean. So we're not alone. Everybody is working on this. This is a very hot topic, an important issue that I think VS is trying to get up-to-date information. And I'm going to leave it to Dr. Moyeno for acknowledgments.
Okay.
So that concludes the portion of the webinar presentation. At this time, we would like to acknowledge all speakers that presented today. Thank you so much. We could not have done this without you. I would like to thank Denise Bonilla, our VS entomologist, for putting these slides together and actually being the lead on this Heartwater exercise. And we really look forward to working with her on future exercises now coming up in June. As far as the National Training and Exercise Program, I'd like to thank Liz Clark for getting this webinar together and all of her staff. And we'd like to thank Dr. Tyler McAlpin from the National Planning and Incident Coordination Group for all of his assistance in getting these tabletop exercises ready. So thank you so much to everyone that's been working on this. And with that, our last slide for the webinar today is any questions.
Ladies and gentlemen, as we move to Q&A, please press #2 on your telephone keypad to enter the question queue. You will hear a notification when your line is unmuted. At that time, please then state your name and question. Once again, pressing #2 will indicate that you wish to ask a question. To submit a written question, please use the chat panel on the right-hand side of your screen and choose All Panelists from the Send To drop-down menu. Thank you.
We do have a couple written questions. The first one is there a public website or page where info on amount of surveillance for all ticks, either by type and or by geographical area? Is there something like that located?
This is Beto. As far as I know, the answer is no. What we can provide is information on surveillance exercises or updates on surveillance efforts, as the one noted by Dr. Soltero there, by the colleagues with CRAD primarily, where they updated on that in 2010. There was another publication, the one we mentioned in 2017, but one page where all this information, excuse me, is compiled or resides, I'm personally not aware of. And I think that may also be one of the efforts that the colleagues with Caribet have been pursuing through their focus now on ticks and tick-borne diseases in the region. The other thing I wanted to mention really quick, since I had the opportunity here, is that there is also a One Health aspect to the tropical bont tick, Amblyomma variegatum, because it can serve as vector of Rickettsia africae that can affect humans. And based on the previous comment before, we understand, and there's a publication by Dr. Sonnenschein in 2018 showing that it appears that the geographic range of Amblyomma americanum in the continental U.S. is expanding, where, now in the U.S., although maculatum was originally limited to the southeastern states bordering the Gulf of Mexico and the South Atlantic states, it appears that that range is expanding northward into the mid-Atlantic states, in the Midwest into Arkansas, Oklahoma, Kansas, and southwestern Tennessee, and even as far west as southern Arizona, and this is for Amblyomma americanum. Thank you.
We have another question. Are Amblyomma ticks obligate biological vectors for the Ehrlichia ruminatum, or can other tick genera serve as vectors of the disease?
Dr. Miller, you want to do that?
So, from what I've read, only Amblyomma are vectors of the disease. Now, in Africa, there are 4 or 5 different species of Amblyomma that transmit the disease. But in the U.S., there's been competency experiments with the Amblyomma that occur in the United States, and of the Amblyomma that occur in the United States, only Amblyomma maculatum that's endemic to the United States, it will transmit the disease. Amblyomma americanum and the other Amblyomma, the Amblyomma americanum, Amblyomma cajunense, Amblyomma imitator, they don't transmit the disease. So luckily, we only have one other Amblyomma that appears to be a competent vector here in the United States. Unfortunately, that Amblyomma maculatum has a fairly broad range in the southeastern United States and may be spreading depending on how global warming affects our climate over time.
Thank you, Bob. This is Beto again, and what I said before on the expansion of the ranges for Amblyomma maculatum, you're correct. Thank you.
We also had a few requests. This is for all speakers. They want to know if the slide deck can be made available.
This is Nolly, and yes, Liz, we can make it available.
Okay. So do you want them to email you, or do we want to just wait till we get the report from WebEx and we'll send it out using those email addresses? I can take care of that.
Yeah, that would be bad.
I'll take care of that.
Thank you.
Okay. At the moment, we don't have any questions in the verbal queue. I don't see any other questions, written questions. We are running a little bit over our time slot, so if there aren't any more questions, we'll go ahead and wrap up. I would like to thank all of you for joining the webinar and let you know that we have another webinar coming up on Thursday, June 14th at 11 a.m. Jan Archer, a pork producer from Eastern North Carolina and the past president of the National Pork Board and a current member of its board of directors, will be the speaker. And the webinar is going to provide an overview of the swine industry and discuss the challenges that pork producers will face in the event of a foreign animal disease outbreak. So I hope you can join us for that webinar also. And as always, if you have any ideas for other webinars, please feel free to contact me. And have a great afternoon.
Thank you for joining today's presentation. The event has now concluded, and you may disconnect.